Hello everybody, and welcome back to BioSC 140 Human Physiology. This is the video for the experiment and data collection portion of Laboratory 6. In Laboratory 6, there's two distinct parts. There's the DNA extraction part, where we're going to use a strawberry to extract DNA, or we're going to extract DNA from a strawberry. And then there is the RFLP analysis and electrophoresis. It's restriction fragment length polymorphism. We'll get into that. All right, so these are the protocols for the DNA extraction within your laboratory supplement. Let's go over it in a little more detail with some photos to go along. So step one is obtain a strawberry and remove the green sepals. The green sepals are the green part on top of the strawberry, and then place it in a Ziploc bag, get the air out of the bag, and then mush it up. We need to break the cells, we need to break it down, we need to turn it into a mush so that we can extract the DNA out of the strawberry. After you have turned your strawberry into a mush, we're going to add 10 milliliters of extraction buffer to the Ziploc bag, and we're going to mash again for an additional 1 minute. This extraction buffer has a few different components. One of the components are salts, and these salts are going to help stabilize the DNA molecule. Another component of it is there's a detergent within the buffer. A detergent is basically a soap. Soaps have molecules that are called amphipathic molecules. Amphipathic molecules have significant polar and significant non-polar portions. These amphipathic molecules, what they'll do is they'll rupture the cell membrane of the strawberry cells and also rupture the nucleus of the cells, allowing the DNA to move into the solution. We are then going to take this buffer and mush strawberry and buffer mixture, and we are going to filter it through a cheesecloth, like as seen down here at the bottom of the screen. We're then going to collect all the liquid that goes to the cheesecloth, and we're going to pour 2 milliliters of the filtered contents into a clean 15 milliliter tube. We are then going to add 5 milliliters of cold ethanol. Now, in class, we have an ice chamber in the back of the room with some ethanol in it, or a jar of ethanol on ice. And when you add the ethanol, it's important that you add it really, really slowly. You're going to have this test tube right here, which has the buffer and strawberry, the filtered buffer and strawberry mixture. And you want to add in the ethanol really, really slowly. What you want is you want the ethanol to sit on top of the mixture. You do not want the ethanol in the mixture to really mix. You want there to be two distinct layers, an ethanol layer and a buffer and strawberry layer. So do not mix the layers. And then what you're going to do is you're going to take a very thin dowel, a very thin piece of wood, and you're going to push it, move it into the test tube. You're going to move it through the ethanol layer and into the buffer and strawberry layer, and you're going to slowly rotate the rod right at that interface between the ethanol and the strawberry mixture. And what you're going to see is you're going to actually see the DNA strands start to spool around. It'll be like a ball of yarn kind of collecting on the end of your stick. And that is your DNA. Right there. So see this student right here? I'll actually start in the top picture. So this top picture, the bottom you can see is the strawberry mixture. On the top, we have our ethanol mixture, our ethanol layer. This top photo has probably been sitting there for a while. All those little imperfections, little like bubbles and stuff, all that is DNA that's been pulled out of solution. If there was no DNA in this top portion, it would be completely clear. So this has been allowed to sit for a while. Down here, you can actually see this kind of mucus looking like substance hanging off of this piece of wood right here. And that's the DNA all spooled together like yarn from the strawberries. The student put the piece of wood into this mixture. You can see the two distinct layers. She moved it around right at the interface of the two layers. And that is the DNA that collected on the piece of wood. The second part of this experiment is the RFLP analysis gel electrophoresis. So we're going to do a gel electrophoresis. Now, these used to be done a lot in scientific labs. I mean, they're still done, but not as often as they used to be done. And what a gel electrophoresis is you have this gel. It's actually a lot like Jell-O. It's basically a piece of, it's a lot like a piece of Jell-O. And what you do, long story short, is you put samples in this piece of Jell-O and you run electricity through it. And the samples you put in will then either move towards the positive side or the negative side, because electricity is flowing through it. And that sample will be separated based on a few different characteristics. So in our lab, what we're going to do, well, we're doing this with DNA. So we have this watery chamber. It's a salt water chamber. It's going to have some buffers, some salty water in it that's going to allow electricity to flow. Pure water doesn't allow electricity to flow through it well, so we added some salts to the water to make it so that the electricity flows better through it. We're then going to put this piece of this gel, it's like Jell-O, inside the water, inside the chamber, and we're going to cut little tiny wells in the gel. Little tiny wells, little holes in one side of the gel. And then we're going to put our DNA molecules, our DNA samples into those wells. Now, DNA is negatively charged, so it's going to move towards the positive side. So we want to make sure our wells are on the negative side so they have, they don't just slip right off the gel really quickly. They have a long ways to go. And after we have this all set up, we have our samples inside the gel, we're going to turn the electricity on. And the electricity is going to flow through the gel from the negative side towards the positive side, and it's going to pull the negative DNA towards the anode side across the gel. Now, when we do this, all right, so that is the procedures. That's how we're going to get our data for this lab. I'll be back in our next video with the discussion of the science behind both of these labs. Or this is one lab, but the two parts of this one lab.